Our next panelist today is Dr. Kelly Drew, and she's going to be talking about hibernation and neuroprotection. So when you're ready, Dr. Drew, you can share your screen and the stage is all yours.
Well, thank you very much. Do you see my screen?
Yes, we can, and we can hear you.
All right, well, thank you for the invitation. It's really a pleasure to participate. I particularly appreciate Dr. Martin's talk. She really set the stage, and so I'll follow up on that. I'm going to describe where our data has led us to the adenosine model of hibernation and neuroprotection. I do have a disclosure financial interest in the company I founded that you mentioned, but I also just wanted to say, I wanted to second Sandy's point about how important it is to make inroads in this area to study the true hibernators and really understand what they do. I'm at the University of Alaska Fairbanks, where I've really benefited from a culture and infrastructure here for environmental physiology, and particularly the work of Brian Barnes and many people who have worked with him here, and myself, who has benefited so much from his understanding of the animals in the wild. And so I'll talk a little bit about that, but I think that that's really important, and that's why so much I appreciate where you're going with this, and I support Trish's effort in this regard. So let's see if I can make my slides advance. Okay, so yeah, I'm talking about hibernation and neuroprotection, and it takes me back to one of our original findings.
Dr. Drew,
yeah?
Could you move just a little bit closer to your computer? Some people are having a hard time hearing you. Just a little closer.
Okay, just let me see if my yeah, that's connected. You should be, let me see if it should be hearing from the, let's see.
Seemed like when you moved closer to your computer, it was a little bit better.
Okay, it should be coming out of my, oh, all right, it should be coming out of my headset, but anyway, I'll try to speak up as well. So anyway, back in 2000, we were curious just about the neurochemistry in the hibernating brain, which led us to use a technique called microdialysis, and we stuck these probes into the animals when they were either euthermic or hibernating. And the most remarkable thing we saw was the difference in the tissue response. And so really in the euthermic animal, you saw reactive microglia and a typical brain injury that would be caused in a non-hibernating animal. But if you looked at the damage in the hibernating animal, it was actually hard to see. And so it really directed my lab towards this understanding of neuroprotection in the hibernating state. And we studied this actually for a couple of decades and still are interested, but in particular, we were looking for something unique about the hibernating state that provided this neuroprotection, and in the process discovered that the Arctic ground squirrel, at least, and I think other, certainly the 13-line ground squirrel and other species, are resistant to ischemia reperfusion injury. They're resistant, they just have an innate species tolerance to injury, and my lab focused on brain injury. And we found that the species difference ultimately had to do with how they handled peroxynitrite and radicals after ischemia reperfusion. So they lose ATP, they depolarize, they release glutamate, but downstream of that, they were able to resist this peroxynitrite. But this was a species effect. And so it didn't explain that difference that we had seen between the hibernating and the euthermic animal in terms of the brain injury. And so it brought us back to this question that Sandy talked about, and to her model that she had proposed many years ago, about the summer-winter switch and the torpor arousal cycle, what regulates those. So our work had really shown that it was the metabolic suppression and the low tissue temperature in hibernation that distinguished the remarkable neuroprotection in the hibernating state. And so we sought to understand both of those switches. And it led us to this adenosine model of hibernation, which I'll describe that it is the winter-summer switch has to do with the increased sensitivity of purinergic signaling. So adenosine signaling is the seasonal switch, which then allows adenosine to really convert sleep into torpor in the torpor arousal cycle. And that's what we see in the winter. So I'll show you the evidence for that. Well, first I'll tell you a little bit about adenosine. So adenosine is a remarkable signaling metabolite. So it's often referred to as a retaliatory metabolite because it's produced as high energy phosphate groups are used up. Adenosine works on 4 different receptors. I'm gonna talk mostly about the A1 receptor because that's what we see seems to be playing a role in hibernation. And tell you that the A1 receptor has 2 really important effects in the brain. One is that it turns down the thermostat. It plays a big role in thermoregulation. Secondly, the A1 receptor is what mediates what we call homeostatic sleep drive. So that's what makes us sleepy when we are sleep deprived. It's why we drink coffee and caffeine, because it blocks both A1 and A2 receptor, but it's the A1 receptor that drives homeostatic sleep. So the other thing that's really remarkable about adenosine is that it is a precursor to SAMe and DNA methylation. So I'll talk about it a little bit more. This is a direction I hope to be able to take in the future about the role of adenosine in epigenetics. And the other thing I want to point out here is this adenosine kinase is a really important enzyme in regulating adenosine levels in brain. And so it provides a sink for adenosine back to AMP, and that will come up later. So in support of this adenosine hypothesis, we have a few observations. And so remembering that adenosine regulates the thermostat in the brain, you can look at body temperature as a readout of central A1 receptor activation. And this is what we see, these remarkable changes in body temperature in hibernation, and not just during torpor and arousal, but this pre-hibernation season is also characterized by a slight decrease in what we call the euthermic body temperature. And this decrease in, say, temperature set point persists throughout the interbout arousals during the hibernation season, and then body temperature comes back in the summer. So there's this gradual decrease in body temperature prior to entrance into hibernation, which we think reflects this winter season switch in adenosine signaling. There's also lower euthermic temperature during arousal. There's these prolonged bouts of torpor, consistent with increased sensitivity to adenosine. And I'll show in subsequent slides that there's an increased sensitivity to A1 receptor agonists, and there's increased sleepiness during the hibernation season. All of these observations support the adenosine model. So this data I show is from free-ranging animals collected by Brian Barnes many years ago. And you can also see in this more recent study this precipitory or this preparatory decrease in body temperature prior to the first torpor bout in the Arctic ground squirrel, free-ranging Arctic ground squirrel. So about 45 days prior to the first torpor bout, you can see a gradual decrease in body temperature. So here's body temperature in days prior to hibernation there. You can also see this seasonal fluctuation in resting euthermic body temperature in the lab. So these are Arctic ground squirrels that were kept in the lab at a 18 degree, kind of a normal room temperature on a 12-12 light-dark cycle. And this illustrates this free-running circadian rhythm that Sandy referred to, that they'll, under constant conditions in the lab, they will still have this circadian rhythm, but the free-running rhythm is a little bit faster than the when they are entrained to the changing light outside. And so you can see their euthermic body temperature waxes and wanes. This is body temperature over about a year and a half cycle, or year and a half time frame. And these are 3 different animals. And so they have this increase and decrease in their body temperature. This is just with pit tags. This is sub-Q temperature. And then you can see that these, during the lulls in body temperature, there drops into torpor. They're not real accurate measures of body temperature, but that's where you see it dropping. And so this is what we see in the lab. And with these animals, then we could test sensitivity to the A1 receptor agonist. Cyclohexyladenosine is the drug we use most commonly as an A1 agonist. And so if you test them at a very low dose, it's 0.1 milligram per kilogram CHA, in animals that are either in the lull of the low point in their annual rhythm of body temperature, or at a high point. When they're in their low point, they respond more. There's a bigger decrease in body temperature in response to this A1 agonist than if you test them at the peak. And so here you can see the significant difference in the maximal change in body temperature after this low dose of CHA. If they're treated in the trough, they have a bigger change in body temperature than if they're treated at the peak. Again, consistent with our model, that there's an increased sensitivity to this A1 receptor agonist and A1 signaling in the winter season. And the same thing is seen when we treat the Arctic ground squirrel to induce torpor with CHA. So CHA, the A1 agonist, when we give it directly into the brain to avoid any peripheral effects, in the summer, here we have rate of oxygen consumption that you can see here, body temperature, what you see here. When we give CHA ICV in the summer, you see a brief gradual decrease in oxygen consumption, a slight decrease, subsequent decrease in body temperature, but it is not sustained. It doesn't progress into torpor. These same animals were kept until the winter season and treated in exactly the same way. And in the winter season, the same dose of CHA, again delivered ICV, produced a significant decrease and persistent decrease in oxygen consumption, with then a persistent gradual decrease in body temperature that looked very much like torpor. So this shows just a little bit more detail. Here is the slide I just showed you in the midwinter season, where CHA induced what looked like torpor, and this is compared to spontaneous entrance into torpor. Looks very similar, and we defined this as an A1 mechanism by comparing an A3 agonist that had no effect, followed by CHA as a positive control, produced a similar response as we'd seen previously. We also can show that the A1 receptor, but not the A2A receptor, is required for torpor, because during spontaneous torpor, again, we've got body temperature, here we have time. If we give the A2A antagonist, it does not reverse spontaneous torpor. But if we give the A1 antagonist, CPT, during onset of spontaneous torpor, it reverses it. So we say that the A1 CNS A1 receptor is both necessary and sufficient to drive onset of torpor in the Arctic ground squirrel, but it has a seasonal sensitivity. So it increases the sensitivity to this drug during the winter season. And in fact, the necessity of the A1 receptor has been demonstrated not just in the Arctic ground squirrel, but also in other types of animals displaying torpor, in the hamster, where the CPT will reverse onset of spontaneous hibernation, and in the fasting-induced torpor in mice, where an A1 antagonist will reverse fasting-induced torpor. So we also see this increased sleepiness consistent with increased A1 receptor signaling in the winter season. This was work done in Craig Heller's lab back in long time ago, published in 1980. But you can see in these golden mantle ground squirrels that, so they just monitored EEG over 2 years and then quantified the duration of sleep. And so percent sleep is here, it's high during the winter months of November, December, January, and lower in the summer months of June and July. And back again high in the winter season. So again, consistent with increased sensitivity to A1 receptor signaling that is the mediator of this homeostatic sleep drive. So we were interested in the seasonal response to CHA to see if it was engaging and having a seasonal difference in either the sleep circuits or in the thermoregulatory circuits. And we really found it's thermoregulation that seems to play a primary role. And so we saw that CHA, this is just using CFOS activation following CHA when it was administered to summer or winter animals. And so we saw, I don't show you here, but we saw activation. CHA definitely activates neurons in the NTS, where others have shown, Domenico Tupone has shown that that's sufficient to induce this effect in rats. And also we see other sleep centers that are activated. But the seasonal difference was seen in 2 areas, one of which was the raphe pallidus. And so the raphe pallidus is the sympathetic premotor neurons that drive thermogenesis. And you can see in this nucleus that activation in the winter is suppressed following CHA, which suppresses that thermogenesis that allows animals then to enter torpor. And so we really see an important role for thermoregulatory pathways. But what could be regulating this seasonal difference? Well, we see these seasonal changes in plasma adenosine. So during this pre-hibernation period, we see an increase in plasma adenosine. This was blood that was just sampled in these animals when they were euthanized for tissue collection, and the metabolomics was run by University of Colorado. And so we see adenosine accumulate, increase in plasma, and then decrease during torpor. This has also been seen by others. But how does it get to from the blood to the brain? We think it enters through these cells called tanocytes. So tanocytes are key metabolic regulators. They're a very unique type of cell that are found in discrete regions, very small regions. One of them is in this, they surround the third ventricle and occupy the median eminence in the hypothalamus. And they serve as barriers between blood and brain. They also produce deiodinase enzymes that regulate the thyroid influence on metabolism. But these cells, we found, have adenosine kinase. So in the adult brain, in astrocytes are typically the cells that express adenosine kinase. In adults, you don't see it in neurons. And astrocytes are really recognized as regulating adenosine levels through expression of adenosine kinase. Whereas if you have more astrocytes and more adenosine kinase, you have lower adenosine. And so we didn't see a seasonal change in the astrocyte and adenosine kinase in astrocytes, but we did see a difference in tanocytes. And so in winter, there's lower adenosine kinase expression in tanocytes. So these cells now that produce a barrier between blood and brain have lower adenosine kinase and can allow adenosine now to come from the blood into the brain. And we think this is fundamental to adenosine tone in the winter season in brain. Interestingly, you see a lot of the adenosine kinase in the nucleus of these cells, and it could be affecting adenosine levels and having an epigenetic influence. So that's something we're pursuing now. So we also see some changes in sensitivity at the receptor level. This is with some GTP binding of tissue from Arctic ground squirrels, forebrain, hippocampus, hypothalamus. This is a functional response with GTP binding. So you can measure actual efficacy and potency of CHA. So this is log CHA. These are the dose response curves. And when you analyze these curves, you can see a slight increase in potency of CHA in the winter season. Pharmacologically, it's not a huge difference, but physiologically, we think it's significant. You don't see a difference. We don't see a difference in the actual receptor expression in the membrane. So it seems to have something to do with regulation of the receptor, and that we have not figured out. But it is more potent in the winter season. So the model proposes that this kind of spillover of adenosine in these thermoregulatory centers bring the dose response of these adenosine, endogenous adenosine, between inhibition of thermogenesis and sleep closer together in the winter season. So the model is that you've got this spillover. In summer, typically you have a adenosine-driven increase in sleep, and at a greater amount of adenosine, which really isn't seen typically in the summer season, you would have an inhibition of thermogenesis. But in winter, the dose response curves come closer, and so the same adenosine that would normally drive sleep is now engaging the inhibition of thermogenesis, and they merge to transition sleep into torpor in the winter season. So that's our model. And we also see, just in terms of this sleep arousal cycle, that adenosine accumulates during the entrance phase. So if you just look at plasma adenosine during torpor and arousal, it increases during interbout arousal. And if you monitor adenosine over time in a torpor bout, this was done by Sarah Rice with chronically implanted animals with chronically implanted arterial cannula. She could sample blood and again measured at University of Colorado metabolomics LC mass spec, and could see that adenosine is high during entrance and then decreases during torpor. So sufficient to be driving sleep into torpor during these interbout arousal sleep hibernation cycles. So like Sandy referred to these two papers that were just published last month, or I guess 2 months ago, identifying cells in mice that regulate a hibernation-like state or that regulate fasting-induced torpor. Again, these cells are part of the thermoregulatory pathways, but it was fantastic work that really identified these cell types, but the results are very consistent to what we have found. But rather than this approach, we have taken more of a pharmacologic approach, which has a more readily accessible path to translate. And that's what we have been doing in rats, really just taking advantage of this thermolytic efficacy that we've discovered about the A1 agonist CHA, and translating that to non-hibernating species, the rat, to induce, we don't really think of it as a hibernation state, but we do think of it as a therapeutic state akin to targeted temperature management that is achieved through this metabolic suppression. So by inhibiting thermogenesis, we inhibit metabolism, and that then, secondary to that, body temperature decreases. And that in the same way models what works in hibernation. So this is just the tip of the iceberg, but we can, by increasing the dose in rats, we can achieve very much the same effect that we see in ground squirrels. But importantly, in rats, we have to prevent them from overcooling. And so we use a target temperature consistent with what is seen in the clinic, which is between 32 and 34 degrees. This is standard of care after cardiac arrest to cool the body for 24 to 48 hours. 24 hours is pretty standard to protect the brain after cardiac arrest. And so we're using this approach in rats to demonstrate the feasibility for clinical studies. And so this data here shows these rats that are treated with continuous CHA through IV cannulas during the period here shown in the gray bar. And so we first put them at a cold surface temperature. So this is through conductive heating and cooling, and administer the CHA. They quickly decrease their body temperature. They also decrease metabolism, which I'll show you later. And then we regulate that body temperature through surface temperature. So we have to increase the surface temperature to prevent overcooling. And in this way, we can maintain the target temperature within the range that we're looking towards. And you do see a decrease in heart rate, but it's just CHA alone. It doesn't seem that even though this pronounced decrease in heart rate, they don't seem to be have insufficient blood flow. And you can see that from their serum lactate levels. Lactate, CHA versus vehicle, actually go down during the period of cooling. O2 saturation in blood goes up. And so there's no evidence of insufficient perfusion, even though heart rate is so low. But I'll tell you more about that actually in this study. So another approach is to maintain or prevent the cardiovascular side effects that are very well known with the adenosine agonist by treating with a peripherally acting antagonist. So we use 8-SPT, is a theophylline derivative. It's a non-selective adenosine receptor antagonist, and can combine that with CHA to reverse the cardiovascular side effects without interfering with the thermolytic effects, which is a centrally mediated site of action. So here's a cardiac arrest model in rats. So it's asphyxial cardiac arrest that we monitor through mean arterial pressure. So you can see it goes down to about 0. And then we treat those animals either with a normothermic control group or in a therapeutic hypothermic group given CHA and 8-SPT. And so we decrease body temperature in that way. And you can see that the normothermic control group, 2 out of 3 animals died before the end of the study at 7 days. And in the therapeutic hypothermia group, all 7 survived, so we could get the brain tissue and look at the histopathology. And for the most part, you see healthy neurons in those animals who were cooled. But in the normothermic control animal, the one that survived, you see these dead neurons. And so, as would be expected, cooling with this mechanism is protective. But we think that it has immediate applications and advantages in the clinic because it targets metabolism, and in that way, the body doesn't fight the cooling, and there are many benefits to that. But sir,
Dr. Drew, unfortunately, we are really running out of time, and we would like to have at least one question
okay,
for your section.
Let me jump ahead then and just say that we can enhance the seasonal sensitivity with dietary restriction. You see a decrease in metabolism. And then just in the conclusions, the data are consistent with the adenosine model. And with that, I can stop and say thank you and have time for a question.
Thank you very much, Dr. Drew. I think we have time for one question, and then we'll save the rest of them for the end of the day during the panel, and we can have those ones later on. But I think Dr. Sherman, we have time for one question.
Yeah, we have a couple here. I'll pick one. So is A1AR, would you consider it a biomarker of torpid states? And is it significantly detectable, if at all, in the blood?
Yeah, so I think that it is a biomarker for both the seasonal preparation prior to torpor, and I think it's a biomarker for the entrance into torpor, from interbout arousal to entrance, and we do see that in blood, which is surprising because we think that it's actually acting in the brain, but it is likely originating in the blood.
Interesting. Okay, thank you very much.
Thank you very much, Dr. Drew, for the great presentation. Our next panelist is Dr. Matteo Seri from University of Bologna in Italy, and he's going to be talking about hibernation and radial protection. So Dr. Seri, when you're ready, you can share your screen, and the stage is all yours.